Today we are going to discuss about the chromatin. As such, there are actually this chromatin material, which is in the condensed form, are at its physiological pH or in its physiological state. This chromatin is divided into two main categories, or we can say the chromatin is divided into euchromatin and the heterochromatin. Euchromatin and the heterochromatin. As we all know that each chromosome, single chromosome, I'm talking about the each chromosome contains a long, very long duplex DNA. It's a single, if we will open up our DNA, it will form a single string, and this string is packed onto the histone proteins. Then all together they form the nucleosomes, then the chromatin material. So it has been observed that there is 5 to 10 times more condensation in mitotic chromosomes than in the interphase chromatin. जब हम लोग इंटरफेस स्टेज में क्रोमेटिन को स्टडी करेंगे, वो लूजली बाउंड होता है, एक चीज। सेकेंड चीज, उसमें लाइट और डार्क स्टेन या लाइट या लाइट रीजन छु बेश ब्याक साइड आसान डार्क रीजन. But when we will compare this our interphase chromatin material with the mitotic chromosome, there is the 5 to 10 times more condensation than the interphase chromatin material. But the nuclear content in the cell is not always in the condensed form. But when there is the two cell cycle processes, like either the meiosis or the mitosis has to take place, that time our DNA material gets completely condensed up to form the chromosome. हिंदी मतलब उर्दू का तर्जुमा है कि ये जो हमारा डीएनए मटेरियल है वो हमेशा कंडेंस्ड फॉर्म में नहीं रहता है, क्रोमोसोम स्ट्रक्चर में नहीं रहता, वो हमेशा इंटरफेस स्टेज में लूजली बाउंड होता है. तूत कॉम्पैक्टली अरेंज्ड आसान के यम साथ सेलस में ज प्रोसेस गसान, अक्ष गसान मियोसिस और दूसरी होती है माइटोसिस. इन दो टाइम पे क्रोमोसोम ज्यादा से ज्यादा कंडेंस हो जाता है, एंड कंडेंसेशन इज अप टू 5 to 10% मोर देन द इंटरफेस स्टेज. ठीक. लेकिन जो हम लोग अभी ना वी हैव टू स्टडी इट्स फिजियोलॉजिकल और हाउ क्रोमेटिन व्हाट आर द डिफरेंट रीजन्स व्हिच आर प्रेजेंट इन द क्रोमेटिन, लाइक आई सेड अर्लियर, it's euchromatin and the heterochromatin. Now we are having advanced technology, like we are having high resolution microscope, or we can use like in case of high resolution microscope we can use fish fluorescent in situ hybridization staining technique, जिसकी वजह से हम लोग molecular mechanism या molecular at a molecular level we will be able to observe a chromosome. Or we can use live imaging. Live imaging कहाँ देखी जाती है? There we can use phase contrast microscope. Maybe in next sem you will be मतलब you will study the microscopy as well inshallah. So phase contrast microscope में क्या होता है? मतलब जब हम लोग light pass करते हैं, due to the diffraction and the refraction mechanism, there is a contrast creating between the cytoplasm and between the different organelles present inside the cell, due to which we will be able to observe live image of a cell. जब एक सेल लाइवली होती है वहां पे जो हमारा साइटोप्लाज्म है सेल का, वो उसमें स्ट्रीमिंग दिखाई जाती है. मतलब जो मतलब अमीबा कैसे स्ट्रीमिंग मूवमेंट दिखाता है, like similarly if we will take a plant cell or the live animal cell under the phase contrast microscope, there we are able to observe the streaming movement of the cytoplasm. सिंपल टेंस सेंटेंस से बोलेंगे, फेज कंट्रास्ट माइक्रोस्कोप के जरिए से हम लोग सेल का जो स्ट्रक्चर है, इंटरनल स्ट्रक्चर, वो ऑब्जर्व कर सकते हैं. एक ऑर्गेनली दूसरी ऑर्गेनली से रिफ्लेक्टिव इंडेक्स के एतबार से वैरी करती है. Even we are having another research, another microscope that is called as fluorescent in situ hybridization. उससे भी हम लोग स्टेनिंग टेक्निक है ये, जिससे हम लोग देख सकते हैं क्रोमेटिन मटेरियल के कौन कौन से टाइप्स है, या जो हमारा क्रोमेटिन मटेरियल है वहाँ पे कौन सा रीजन कैसा है. ठीक है. तो जो ये हमारा सेकंड यूनिट है, दिस सेकंड यूनिट इज ऑल अबाउट द क्रोमोसोम ओनली. ठीक है. हमें इसमें क्रोमोसोम का ही पढ़ना है. और ये इजी है, छोटा है, ज़्यादा टफ नहीं है. Now if we'll talk about the chromatin material, as I said, it's having two regions, euchromatin and the heterochromatin. If we will say what is euchromatin, simply Shaisana, euchromatin is the active part of the DNA and heterochromatin is the inactive part of DNA. But while we stain our nuclear material, there we observe some light bands and some dark bands. As you can see in this, mm, you can come on the whiteboard where I will draw the assume that this yellow line is our simple nuclear material. But somewhere we are observing that the some region on this DNA is darkly stained and the some region is lightly stained. So the lightly stained region we called this as euchromatin and the darkly stained region we called it as heterochromatin. Okay, again I will repeat. When we stain our nuclear material, then observing this nuclear material under the microscope, we observe light and dark banding staining pattern on this nuclear material. The light, the part of the DNA which is lightly stained is called as euchromatin, and the band and the part or the region on the DNA which is darkly stained is called as heterochromatin. So first explanation for why this euchromatin stain lightly, because the DNA content or the DNA material present in the euchromatin is loosely packed. यूथ इस टाइट टाइट चीज था टूथ रेड से डार्क स्टेन, यूथ इस लूज लूज चीज था टूथ रेड स्टेन बराबर, मगर सो दूर वाज लाइट स्टेन ना. Similarly, heterochromatin में क्या होता है, there is a tight packaging of nuclear material, so that's why they stain dark. This is the these are the first two like differences between the euchromatin and the heterochromatin. And in most of the cases we always ask about the differentiate between euchromatin and the heterochromatin part, so you should be able to describe each and every point. Why if it is darkly stained, why this is darkly stained, if it is lightly stained, why. And the region of the eukaryotic chromatin material is found to be GC rich. जो ये हमारा यूकेरेटिन जो पार्ट है हमारा, दिस इज द जीसी रिच कंटेंट, मींस इसमें जो हमारा एटीजीसी जो बेसिस है इसमें सबसे ज्यादा देखा गया है जीसी रिच, इसमें बेसिस होती है. अच्छा, हाउ मेनी बैंड्स आर फॉर्म्ड इन जीसी रिच एरिया. जीसी के बीच में जो है ना, साइटोसिन एंड गुआनिन के बीच में जो पेयरिंग होती है उसमें कितने बैंड्स बनते हैं, एनीवन. हाँ, ठीक हो गया ये. एंड द दिस इज जीसी कंटेंट रिच एंड द क्या बोलते हैं, एंड इन केस ऑफ हेट्रोक्रोमेटिन इट हैज बीन ऑब्जर्व दैट दिस डीएनए इज हैविंग 80 रिच कंटेंट. दिस इज द थर्ड पॉइंट. जो हमारा यूक्रोमेटिन है दैट इज जीसी रिच रीजन, दे where cytosine and guanine are present in higher concentration. And in case of heterochromatin, this is 80 rich region. Or in other words, we can say this euchromatin hai, this is transcriptionally active. This euchromatin contains those genes which are transcribing every time, which are getting transcribed and translated every time, means transcriptionally active. और जो हमारा ये है heterochromatin, this is most of the times this is transcriptionally inactive. This is transcriptionally inactive. And the genes which are present in euchromatin are always getting transcribed. But the genes which are present in heterochromatin region, there are sometimes that some genes are getting transcribed. There is a मतलब ताशु fluctuation गसान अक्चु आसान जो heterochromatin है, that is again into two types. We will discuss that thing also. So this is about the basics मतलब chromatin and heterochromatin. Now if we, as I said right now, there are some genes which are getting transcribed in heterochromatin region. It means there is some part is fluctuate through gasan heterochromatin to euchromatin, or sometimes again this euchromatin, due to some modifications, is getting converted back into the heterochromatin. So again, when we studied all the heterochromatin, what is happening in the heterochromatin? It has been observed that the heterochromatin, वहाँ पे heterochromatin में क्या होता है? Heterochromatin is the region where we have found the repeated sequences, where we have found the repeated sequences. Or the repetitive sequences, these are mostly at the centromere and the telomere side. जो ये heterochromatin part है chromosome में, these are near to the centromere and the telomere region. Again, it has been said that there are some regions present on the heterochromatic DNA material that always remain in an inactive form, that never get transcribed. Transcribed मतलब को DNA to RNA बनाओ, replication को बदल चीज DNA to DNA बनाओ. तो we are talking about the formation of RNA from the DNA, that is called as transcription. So the DNA material which is never getting transcribed is called as constitutive heterochromatin region. Constitutive heterochromatin region. And there is another category in this heterochromatin which gets converted to euchromatin when there is a need. Then again, this euchromatin, when there is no need of that transcription, gets converted into the heterochromatin. And this type of heterochromatin is called as facultative heterochromatin. That you have heard about the facultative microorganisms, where which may or may not depend on the oxygen. तो similar concept आप यहाँ पे याद रखेंगे. Heterochromatin is a region which is able to get sometimes get translated into the transcribed. So if we will talk about the specific, what are the specific points which we can talk about this constitutive heterochromatin, मतलब वो जो हमेशा permanently heterochromatin रहता है. First we can say it is permanent or it always remain in a condensed form. जो हमारा constitutive heterochromatin है वो क्या है? That is the permanent or it always remain in the condensed form. This is the first peculiarity about the heterochromatin. अब हम लोग उसमें किसकी बात करें? Constitutive heterochromatin की. And this constitutive heterochromatin replicates in the late S phase stage of cell cycle. You see constitutive heterochromatin ये क्या ये जो permanently रोज़ान heterochromatin ये छुन गसान shift किए वो ये जो permanent रोज़ान बेच you most of the times रोज़ान condense. Second has भी वाना I'm second part में बोल रही हूँ ये replicate कब होता है? Late S phase stage. ठीक ये tightly packed है. So it means when there is मतलब like less chances of genetic recombination इसमें नहीं होती है क्योंकि जो tightly packed अच्छा space ही क्या है कि ऐसे कैसे crossing over. So it means जो हमारा ये है constitutive heterochromatin region, there is a reduced frequency of genetic recombination. There is less frequency of genetic recombination. और जो ये हमारा constitutive region heterochromatin region है, they are not getting transcribed. Uh, Next हम लोग बोल सकते हैं this region is consisting of multiple repeats. जो ये है ना ये जो region होता है heterochromatin ये अक्सर किन का होता है? इनमें multiple ये होता है, multiple repeats होता है इनमें present. So this was about the constitutive heterochromatin. Now I said one thing about that there are some changes which takes place between the euchromatin and the heterochromatin. It means DNA में कुछ ऐसी changes होती है जहाँ पे DNA convergence होते हैं from U to hetero, hetero to U. But we have used certain molecular markers by which we were able to observe the property of the DNA, where we have observed that there is a modification in the DNA and sometimes there is a modification in the histone proteins which allow this conversion. Bhavan, again I will repeat. जो ये यूक्रोमेटिन है, मैंने एक पॉइंट बोला कि ये हेट्रोक्रोमेटिन में ट्रांसफर होता है या हेट्रोक्रोमेटिन यूक्रोमेटिन में ट्रांसफर होता है। विद द हेल्प ऑफ यूजिंग मॉलिक्यूलर मार्कर्स हमने ऑब्जर्व किया कि चेंजेस डीएनए में कभी हो जाती है या तो कभी हिस्टोन प्रोटीन्स में हो जाती है, जिसकी वजह से टाइटली पैक्ड डीएनए कन्वर्ट हो जाता है लूजली पैक्ड में, या लूजली पैक्ड डीएनए कन्वर्ट हो जाता है टाइटली पैक्ड में। नाउ what are these modifications? First modification occur in the DNA itself. ठीक है, आप ये जो मैं नोट ये बोल रही हूँ ना, these may be not the exact note, whatever I am giving you lecture is your complete note, you can find down by yourself. I have material, I will send you, but still कुछ कुछ changes रहती है पढ़ाने में. So first there are actually the 5 changes which occur during this conversion from U to hetero to U. First itself is the modification in the DNA, first point. Second is the histone modification in the histone acetylation, histones की acetylation process हो जाती है. Third is the methylation of histone proteins. आपकी कसम कोऑर्डिनेट चेंज किसमें हो जाती है? डीएनए itself. Second is the acetylation of histones. Third is the methylation of histones. Fourth is the ubiquitination of histones. And fifth is the phosphorylation of histones. First modification occurs in the DNA, then all the rest of the 4 occurs on the histone proteins. So these processes are acetylation, methylation, ubiquitination, and phosphorylation. These are the 4 processes which occur during this conversion. So if I will talk about the first modification which occur on the DNA is the whenever, you can come on the screen again, we are having assumed that we are having like we are having GCCG. We are having the sequence on the double stranded DNA. It has been that when the C is first to the G, मतलब वो जब साइटोसिन G से पहले होगा उस साइट पे इसकी मिथाइलेशन होती है, या हम लोग लिखेंगे GGC क्या, ठीक, ये हमारी समझो, ये हमारी सीक्वेंस है. व्हाट आई सेड, वेन एवर साइटोसिन इज प्रेजेंट बिफोर द ग्वानिन, जहाँ पे साइटोसिन पहले होगा ग्वानिन बाद में होगा, उसकी मिथाइलेशन होती है. दिस इज द फर्स्ट चेंज विच ऑकर ड्यूरिंग दिस ट्रांसफॉर्मेशन. सो जो हमारा ये सी वाला है इसके नेक्स्ट क्या होना चाहिए था, जी, तो इस साइटोसिन की मिथाइलेशन नहीं होगी. तो मिथाइलेशन का कॉन्सेप्ट क्या है, जब साइटोसिन के बाद गॉनिन होगा तब उस साइटोसिन की मिथाइलेशन होती है. Again in the lower case we will see, again we are having यहाँ C है, we are having 2C, first is this C and second is this C. So but in first C we are having next guanine, so methylation will occur here. Then again we are having second C, but to this second C we are having adenine. So the criteria is we should be having guanine there. So no methylation will occur on this cytosine. This is the first modification which occurs on the DNA. So second modifications are like acetylation, methylation, ubiquitination and phosphorylation. So now how modifications occur? They include reduced methylation of histone proteins. First क्या होता है? Reduced methylation of histone proteins, या तो increase in methylation at particular site on histones. Methylation of histone proteins हो जाता है first, reduce methylation of histone proteins, or sometimes increase methylation of particular site on histone, or we can say there is a methylation of cytosine bases in the DNA, or there is the methylation of cytosine base in the DNA. All these processes work together leads the transformation of physiologically state DNA from euchromatin to heterochromatin region. So this is how the transformation takes place. Scientists have carried one मतलब like they have observed one territory जैसे इंडिया का है ना मतलब कौन सा स्टेट किसके साथ है कहाँ पे क्या चीजें होती है साइंटिस्ट ने एक सेल में we are having different chromosomes what they have done what scientists have observed they have observed some specific pattern they have observed some specific pattern of chromosomes or we can say overall chromosomal organization within in the nucleus. Again, I will repeat. Scientists have observed or studied that how chromosomes we are having different number of chromosomes, how these chromosomes are arranged within in the nucleus or overall chromosomal organization within in the nucleus. So they have make a three-dimensional space of all the nuclear material present in the DNA and they call this as a chromosome territories as you can see in the diagram first on the top side on the left hand side we are having chromosome number 5 uske baad 6 8 11 then x y chromosome 12 15 13 like this way we are having arrangement of chromosomes inside the nucleus so this is something different which we are learning today so they observed they couldn't establish much about this how there is interaction but they have observed कि एक के वजह से फिफ्थ को कोई प्रोटीन जरूरत है जो सिक्स बनाता है सिक्स नियर बाय है इसके तो सिक्स क्या करता है प्रोटीन अपना देता है जो फंक्शनिंग है फिफ्थ के मतलब ये ऐसे अरेंज होता है कि इनकी जो फंक्शनिंग है वो स्मूथ चले जो इनकी फंक्शनिंग है जो इनको एक दूसरे के साथ रिक्वायरमेंट्स है जो इनको नीड है एक दूसरे के साथ वो ये कॉम्पैक्टेबल तरीके से बनाते हैं एंड इट वेरीज फ्रॉम cell to cell depends on the function of a particular tissue so this is the how these there is a change in the chromosomal territories this is all about the today's lecture euchromatin and the heterochromatin